Thank you to all of our presenters, Dr. Raney, Ramezanli, and Dr. Ghosh. So we're going to go ahead and get started with our question and answer section. So if anyone has questions in the audience, please put them in the chat pod and we will get to as many questions as we can today. And with that, we'll get started with Dr. Raney. Thank you for clarifying that the no difference standard described in product-specific guidances for topical products is essentially based on Q1 and Q2 sameness. The question is, how should a generic drug developer develop a generic product differently depending upon whether we are trying to achieve Q1, Q2, Q3 sameness versus Q1, Q2, Q3 similarity?
Okay. That's a really good question. So for you, the reality is you would treat it no differently. You would do your best to be identical to the reference product. So you would want to characterize all the complexity of the reference product, to deformulate it, and really you're aiming to be Q1 and Q2 the same, to be as identical as you can. And when you have done that or done the best you can, and you've characterized Q1, Q2, and Q3, let's say that you've demonstrated that you think you've matched it really well, then send that in to the FDA. You can always come in a pre-ANDA meeting. And once you've put your formulation composition together, FDA will assess it based upon scientific principles of Q1, Q2, Q3 similarity, or really the no difference standard. Not so much based upon, what you don't have to worry about is that you're going to be rejected on some regulatory technicality. What this essentially helps to ensure is that we're really focused on the scientific validity of that formulation and how well it matches the reference product. So that's assuming that you've matched it really well and you can show that you've matched it really well. The other scenario is that you've done your best to match it, but you can tell that there are or appear to be some differences between the reference product and your prospective generic product. So here, in scenario 2, you actually know that there are some differences. In this situation, the question is, are these differences that are going to affect local or systemic bioavailability? Are they acceptable differences? So in this situation, you would communicate with FDA. You would ask whether this is acceptable. Perhaps FDA might already have evidence that that particular difference would not affect local or systemic bioavailability. Or alternately, so that's either evidence already available to the FDA, or you would need to submit evidence in your ANDA. For example, you might propose to perform an in vivo DOFM study like Dr. Ramezanli talked about, in which you'd be measuring the local PK as well as monitoring the systemic PK in that study, and thereby you'd provide evidence that the particular differences you've characterized between the test and reference products do not affect local or systemic bioavailability.
Great, thank you so much. Our next question is for Dr. Ghosh. Does Raman spectroscopy allow one to discriminate metabolites or degradation products relative to apparent?
Hello. Thank you so much for the question. I think it really depends on what peak or signal we are tracking for to track the molecule in the skin. For example, with cyanophenol, when we were essentially using the signal from the carbon-nitrile bond, if that molecule were to metabolize in the skin, and if we didn't have an unique signal associated with the metabolite itself, then it may be challenging to track. However, with other molecules where we have unique bonds in both the parent as well as the metabolite, it may be possible to use techniques such as Raman to independently track the molecule, independently track the parent and the molecule in the skin. Thank you.
Thank you, Dr. Ghosh. Our next question is for Dr. Ram. For Sam, can we use the designated RS product instead of the RLD product as a reference for deformulation and for Q1, Q2, and Q3 matching? Dr. Raney.
Thank you. Yes, the short answer is the designated reference standard product can be used for the deformulation and for the formulation comparison and assessment. This is a good question because unlike products that are regulated, required really to be Q1, Q2 identical, in which case every generic that's approved is Q1, Q2 identical to the RLD product, and then even if one of those becomes designated as the RS, it would still be the same. That's not true for topical products. So sometimes a generic product that becomes the reference standard may have a different composition than the RLD, and it is acceptable to use that as the reference, as the designated reference, so the RS can be used in that way.
Thank you, Dr. Raney. Our next question is for Dr. Ramezanli. How can we get confirmation from FDA related to the acceptability of our test product's formulation composition for an alternative BE approach using DOF?
Hello, thank you for this question. So since these PK-based approaches using microperfusion and microdialysis are considered novel approaches for demonstration of bioequivalence for topical dermatological drug products, if you want to pursue one of these approaches, we highly recommend you to submit a product development meeting request to the agency in which you would include your proposed test formulation and your proposed product development plan, proposed study designs for both the method optimization studies and the pivotal studies, and any preliminary data that you may have to justify the study design that you are proposed. And as part of reviewing this meeting package, we would evaluate the eligibility of your proposed test formulation for that specific BE approach.
Great, thank you. Our next question is for Dr. Raney. How are the Q2 sameness tolerances of a plus or minus 5% set? Why not plus or minus 10% or 20%, or why not 1% or 2.5%?
Okay, that's a fair question. The answer is historical. So it predates my time, but my understanding from when I've asked this question is that at the time the question was posed of many people in industry who were manufacturers, developers, drug developers, and the feedback that came back, the consensus appeared to be that if an individual ingredient had a variation of plus or minus 5%, that was likely within manufacturing tolerances, and it was unlikely, in the opinion of those experienced pharmaceutical professionals, that would be that would make any difference in the product. Now, the reality is, this is a question that, because it's so keen to the way we regulate products now, that we are assessing scientifically. So we have research in this area to assess what happens when one goes beyond plus or minus 5%, and is it that plus or minus 10% or 20% may be acceptable, but more importantly, it may not be the same for every ingredient. And specifically, it may be the thermodynamic activity of the product that we really need to monitor. So some differences may affect the solubility of a product, even if they change by plus or minus 10%. Other differences, they may not affect the solubility, they may not affect the thermodynamic activity, may not affect the bioequivalence. So this is an area of active research for us.
Thank you, Dr. Raney. Our next question is for Dr. Ghosh. If the reference product appears to utilize a proprietary excipient, how should a generic drug developer present the Q1, Q2 information for that excipient in a composition?
Thank you for the question. That is actually a fantastic question. So our expectation is when a prospective generic manufacturer is looking at matching formulations to essentially utilize it as part of characterization-based approach, that within the scope of product development, the applicant would need to deformulate the reference product and thereby identify not only the type of inactive ingredient in the reference product, but also the amount of each of those inactive ingredients. And the agency's current expectation is when you provide such information to us in either a control correspondence or a pre-NDA meeting package, you should include the information about the inactive ingredient that you are proposing to use for the proposed generic product, along with the relevant compendial monograph, if applicable, as well as the amount to 2 decimal places as discussed yesterday. You can also include the brand name of the excipient if you choose to use a brand name excipient to support the evaluation of the proposed test formulation. Thank you.
Great, thank you. And our next question is for Dr. Ramezanli. Can in vivo cutaneous PK-based BE approaches, such as those using dermal sampling techniques like DOFM or DMD, be used for a demonstration of bioequivalence with a topical patch?
Thank you. That's actually a very good question. Yes, you can certainly use it to compare local bioavailability of a test and reference topical patch or topical delivery system. However, you may need to use alternative designs during the method development to show discriminatory between different PK profiles and show discriminatory capability of your proposed method. For example, a dose sensitivity study that I described for a semi-solid drug product may not be applicable when you have a topical patch or topical delivery system. And instead of that study, you can use different dose duration of application of that patch and compare the bioavailability of the drug when you apply the patch or a topical delivery system for a different amount of time, and show that you can get differences in the bioavailability when you apply the patch within different dose durations, and you can get differentiated AUC or Cmax and use that instead of a dose sensitivity study, for example. Thank you.
Thank you so much. And thank you to all of our presenters and panelists and to the audience who asked such great questions. And with that, that's all.